Hi students, here is the second narration for the topic that we're covering this week, the lymphatic and immune system, and this narration will focus on the immune system's third line of defense. So the third line of defense, in comparison to the first and second line, is specific immunity, and it also has another name. It's called adaptive immunity. What that means is the response by the cells of the third line of defense, is very specific for the type of antigen that the immune response is being made against. So if you've listened to the first narration, you would know that the first and second line of defense are referred to as non-specific. Another word for non-specific is innate, meaning you're born with all of those defenses. Adaptive immunity, which specifically uses as the effector cells B lymphocytes and T lymphocytes, and most often I call them B cells and T cells. These cells can respond to a wide variety of antigens. And one of the key features of adaptive immunity that is not found with the first and second line of defense is that this response will be systemic. It's all around the body. It is specific for the antigen, and more importantly, after responding to antigen, memory T cells are left behind. These are B cells and T cells that remember the antigen to which they had originally responded. So usually when these two cells make our first response, we call that the primary response. And if they meet the same antigen, this could be years later, memory cells can respond to that similar antigen, the same antigen, again. So let's get started. So the two cell types that are the effector cells are B lymphocytes and T lymphocytes. If you remember, these are the agranular type of leukocytes when we were looking at formed elements in blood. And the most important effector mechanism that B lymphocytes use is that they make antibodies. There is no other cell in the immune system that makes antibodies. Only B lymphocytes make antibodies. And pardon me, I could draw a cell here that's a B cell. So let's put a B in it. And an antibody molecule is usually demonstrated as a Y-shaped protein. And it can be, at first, it's usually embedded in the plasma membrane of the cell. And after the cell responds, it can be secreted. So for this reason, if you think back to our study of blood, when we looked at plasma and all of the constituents of plasma, we had ions in plasma. Plasma was a liquid portion of blood, so anything basically that can dissolve in water usually was found in plasma. We also had proteins, and one of those proteins was called globulin. Well, the correct name for an antibody molecule is immunoglobulin. So an antibody is a Y-shaped protein made by B cells, and it has two arms, so to speak. And at the tip of these arms are the sites, the places where the antigen can bind. So an antigen can bind, let's say here, and usually it has to bind both arms. And so this would be the antigen binding. Now the binding site, so that's the antigen binding site, so AG for antigen binding site. That antigen binding site is unique for every single antibody molecule. But we'll get into that next couple of slides. So B lymphocytes then can recognize specific antigens. These are markers, these are substances, molecules on the surface of the pathogen, or it could also be if a phagocyte has chewed up the pathogen, like a bacteria, and kind of, left parts of the bacteria floating around in the bloodstream or in lymph, a B lymphocyte can recognize it and it can use its antibody molecule to bind to it. So the antibody molecule for a B cell is considered the B cell receptor. It's a receptor that binds antigen. And so when we talk about the third line of defense, we're going to look specifically at viruses and bacteria as the, two types of microorganisms. And like I said in the previous slide, bacteria can multiply on the surface of a desk, your telephone, inanimate objects, whereas viruses must enter a cell in order to replicate. So the question is, if viruses must enter a cell in order to replicate, how will the immune system see them? See parts of the virus? Because in this discussion, when we speak of an antigen, we are speaking of a substance that's really, really small. And like I said before, most antigens are proteins, and they're about 9 to 11 amino acids long, maybe 13 amino acids long, not usually more than that. So they're very small. So fragments or parts of these microorganisms can be seen by B lymphocytes if they're free, floating around. They're present somewhere. T lymphocytes, on the other hand, need help to see the antigen. So when we say see, we mean recognize it, be able to bind to it. And that help is provided by another cell type called an antigen-presenting cell. Now, an antigen-presenting cell is not just any cell. There are specific cells in the immune system whose function is to present antigen. So a macrophage, for example, functions as a phagocyte in the innate immune response, but it can also function as an antigen-presenting cell in the adaptive immune response. So T lymphocytes are different from B lymphocytes in this regard. They do not see free floating antigen. The antigen must be presented to them on the surface of another cell. Then they can recognize it. Now, different from B lymphocytes also is that T lymphocytes come in sort of three varieties. They're what are called helper T cells because they help usually upregulate the immune response. They get it going. Cytotoxic T cells, these are like killer whales. They can kill. They can kill another cell. And then we have what are memory T cells that are left behind. So the response in an adaptive immune response, the lymphocytes are systemic. They are found basically in blood. They can be found in lymph because remember, white blood cells can migrate in and out of body fluids basically. So they can be found in the spleen, lymph nodes, tonsils, which are patches of lymphatic tissue found around the openings for the oral cavity, so at the back of the throat, and also the nasal cavity. Peyer's patches are lymphatic tissue found in the large and small intestine. So as it says here, the third line of defense is responsive to a specific type of infection. Now, T lymphocytes and APCs. So what this image is trying to show is, here is the peach colored structure here. That's an antigen presenting cell. And you do not have to know the details or memorize this diagram. But what it's showing you is that within the antigen presenting cell, there is a mechanism by which parts of the microorganism, whether it's bacteria or virus, is presented. So here is the presentation. This is the T cell. And the T cell has a specific receptor also. Just like the antibody is a receptor on a B cell, the T cell has a T cell receptor. And that T cell receptor has to be able to bind to the molecule that is presented to the T cell with the antigen in its groove. So what's sort of barely visible here is the dark green is a molecule that is found on the surface of this, the APC. But it has a groove, and within that groove is where you find the antigen. And the T cell, with its T cell receptor, must bind that entire complex there. So the fit between the T cell receptor and the molecule on the surface of the antigen presenting cell plus, so let's write this out, a T cell is able to see, or it needs to have, a molecule on the surface of the antigen presenting cell. So this would be the APC plus antigen. AG is antigen. That's what the T cell sees. It does not see free antigen. So if a virus can somehow get into a cell and avoid being presented to a T cell, right, and from that cell, then there's no way the immune system will see it. So this is the reason why T cells are critical for viral infections. So let's move on from this slide. Now, what happens when a T cell actually can interact with the antigen presenting cell plus the antigen that's being presented to it? So again, the T cell receptor has to be specific. So it's this lock and key fit. Again, we come back to this, right? Specificity. Only a specific key can open a particular lock, right? Not any key would work. So it's the same thing here. The T cell receptor has to be able to. So here we see the T cell on this side, and this is showing you an enlarged version of what is being presented. So here again, this part is being presented to the T cell. And that presentation to the T cell activates the T cell. So in the bone marrow, we know we have stem cells, and they differentiate into inactive T cells called naive T cells. Now, T cells do go to the thymus for further maturation, and that is going to be discussed on the very last slide for this series of slides here. But right now, what we want to focus on is, here we have a perfectly normal T cell, a mature T cell, a T cell that has a receptor, and it's looking out for antigen being presented to it. But only if there's a good fit between the receptor, this is a physical fit, like a lock and key, only if there's a good fit between the T cell receptor and the antigen in the groove of that molecule present on the antigen presenting cell, only if there's a good fit between the two, then the T cells will become activated. So let's say the T cell can fit, and now this causes activation of the T cell. When we say activation, we mean there's a whole lot of cellular signaling that's going on. There are pathways in the cell that basically gets the cell into gear for doing something. Now, if it is a helper T cell, what the helper T cell does, it releases a lot of chemicals that recruit other cells. It can recruit B cells, it can recruit more T cells. But if it's a cytotoxic T cell, a killer T cell, then what it will do, it will kill the cell that's doing the presentation, the antigen presenting cell. Because remember, that antigen presenting cell is probably infected with the virus, and that's why it's able to present part of the virus to the T cell. So T cells, when they become activated, they will make copies. So this is called clonal expansion, because they will make copies on themselves. So they just go through rapid mitosis, right? A lot of mitosis, produce millions. So what you end up happening is you will have millions of the T cell with the type of receptor that can respond to a specific antigen, the antigen of the virus that's in your body. And that's a good thing, because now you have, it's like you only had, one soldier that can recognize the enemy. Now you have a million soldiers that can recognize the enemy. So it's that idea. So these clones can only destroy the specific pathogen that carries the matching antigen presented by the antigen presenting cell. Some of the activated T cells will become long-lived memory T cells, and that's a really important characteristic of the adaptive immune response. So memory T cells live for years, literally. And I think they've at least been clocked by, 10 years, 12 years. So these memory T cells just stay in the body, and if they encounter the same antigen again, then they quickly become activated faster than in this first response. So the first response is what's called a primary response. Secondary response is when they see the same antigen a second time. Because remember, now that you have clonal expansion, all of these T cells will go to work, get rid of that virus, and you don't feel sick. So the cells that had the virus were killed in the process, but again, because the virus depends on the cell to survive, the killing of the cell means the death of the virus also. So in that way, the T cells help in an immune response. So T cells are critical for the viral response. Now, what about B cells? How do they become activated? So it's a similar principle, except in this case, if we look here, B cells have, so here's a diagram of B cells with antibodies on the surface. Now, in this diagram here, they're using color to convey the concept that each B cell makes a specific antibody molecule. So this one, let's say it's making antibody to the flu virus. This one is making antibodies to COVID. This one is making antibodies to measles virus. Every B cell only makes antibodies of one specificity, meaning the antibodies can only bind one specific antigen. It's an important concept because it means we have to have millions of B cells to make millions of different types of antibodies for all of the millions of antigens that over time and years will enter our body. And also, an antigen, the flu virus may have 20 different antigens that the immune system can see. Because remember, an antigen is anything that is foreign to our body. So as long as it looks foreign to our body, the immune system will respond. So it isn't one virus, one antigen that the immune system sees, it's many. So B cells, like T cells, are activated when they come in contact with antigens. So in this diagram, we're seeing that the antigens are these little red dots that are shown here. And remember that B cells do not need help seeing antigen. They don't need an antigen presenting cell. They can see free floating antigen. So the B cell with the antibody that can respond, bind to the free floating antigen, is the one that is shown here that becomes activated. The others, they just keep moving around in the body, hoping that one day they will meet an antigen that they can bind to. So the one that has the B cell receptor, the antibody molecule is a B cell receptor, that is now activated, that one, or those ones, because there'll probably be a couple, they go into rapid clonal expansion, rapid mitosis. So lots of copies of this exact B cell will be made, millions again. And again, the reason is the same. If this is the antibody that is capable of binding the antigen that is floating around in the body, then you want to have lots of that antibody available to help mop up the antigen. So this is what we're seeing here. So some of these B cells differentiate further to become plasma cells. And a plasma cell is just a B cell that secretes its antibodies. That's all it is. So now we have all of these antibodies in the bloodstream. So this is why we could take a sample of somebody's blood and actually find out, do they have antibodies to, let's say, chickenpox virus? So when we talk about blood plasma having globulins and immunoglobulins and proteins, this is an example of how those proteins ended up there. Some of these B cells, like this one here, they remain behind and they become what is called a memory cell. And the next time that same antigen, and it has to be the same antigen, because remember, the antibody molecule is specific for one antigen only. But if this memory B cell comes in contact with the antigen again, shown here, then it reverts to being a plasma cell and it just makes, again, a large amount of antibodies. So this is the principle upon which vaccination is based. It is very difficult to get a T cell response to a vaccine, but it is much easier if the vaccine is well made to get a B cell response and to get lots of antibodies produced. So when you get the vaccine, this is the response here, the primary response, because that's the first time your B cells will be seeing the antigen. So the vaccine is usually an antigen and portion of the organism. And it could be a virus or a bacteria. But that would be your first response. So you make some antibodies, etc. But more importantly, what you make are these memory cells. And they remain behind for years. So this is why you can wait 10 years before you need a tetanus booster again. If you happen to get infected with the tetanus bacteria, then what the memory cells will do is this part here. They will quickly go into gear and make tons and tons and tons of antibodies to try and protect you. So activated B cells, they bind to the antigen, they produce antibodies, and some of them become memory B cells. Now, cooperation between the innate and adaptive immune response has to occur. So again, we learn, we break down the immune system into these parts, innate, adaptive, first, second, third line, to try to understand the mechanisms that are involved. But at the end of the day, the entire immune system has to work together. So if we get an infection, for example, in our throat, that could be a strep throat type of infection, that's bacterial. Initially, the bacteria has been able to breach the first line of defense, the physical barrier, the mucous membrane. Phagocytes are working in the second line of defense, right? That's why you can actually see sometimes the pus that is there at the back of the throat. But even that isn't enough. Then we have to call in the third line of defense. And so this is where the T cells and B cells will come and try to enhance, right, the overall immune response. So the innate immune system enhances adaptive response so they can be more effective. All three lines must work together. Adaptive immunity is long-lasting and has memory. Often the secondary response produces a quick and stronger immune response so that you recover from infection much easier and faster. So this is what is shown here, and like I said, it's the principle of vaccination. So let's say you got vaccinated here. Or let's say there's no vaccine for this virus that's infecting you, like we had with COVID, that's initial exposure there. And on this side of the graph, we're looking at the concentration, how much antibody is produced. So what we'll see is that for the primary immune response, that's the first time you're seeing this infectious agent, right? It is a longer time, there's a peak in the response, and then usually this line coming down here now, the slope, that means that the concentration of antibody is slowing down, but that's because there's no need for more. You have been able to combat the infection. So you recovered. In the case of a vaccine, well, you probably felt bad a couple of days, but eventually you're back to normal. Now, let's say sometime later, you get a secondary exposure, meaning you actually get infected. So let's say this was a vaccine. Now here, the flu virus has actually entered your body. So here you got the flu vaccine, weeks ago. Now you're out and about and you get the flu virus. What happens? Well, this peak, first of all, is much higher than this peak. So the amount of antibody that's produced is significantly higher. Also, whereas it took this much time before you could arrive at the peak, the time difference here is actually much shorter, usually. So the secondary response is faster, and it's a much bigger response. And that is what helps you to feel better. You actually feel better. You don't feel sick necessarily at all. This is happening in your body, but you're completely unaware of it. So this is the last slide that I was talking about in terms of T cells. And an important feature about T cells is T cells mature within the thymus. So for a gland that is so important, we hardly ever mention it in anatomy and physiology, except when we get to the immune system. And hematopoiesis occurs in the bone marrow. So hematopoiesis is the production of all those formed elements we found in blood. Now, immature T cells, however, leave the bone marrow and they go to the thymus. And when they go to the thymus, they have to go through several developmental stages to ensure that they can both function, so their T cell receptor is good, and tolerance. Tolerance means that these T cells will not attack your own body. So, unfortunately, we have diseases where the immune system thinks that your own tissue is somehow foreign antigens, and it starts to attack the tissue. So rheumatoid arthritis is one example, where the immune response is directed at the cartilage in the joints. Over time, that immune response destroys the tissue, and that leads to disfiguration, of the joints. So that's just one example. So what has happened is both T cells and B cells must learn to be tolerant of what we call self-antigens. Self-antigens are all those molecules on the surface of our cells that belong to us. Because again, all antigens are proteins, and all proteins are dictated by what's in our DNA. So, as long as T cells have a functional T cell receptor, and as long as they have learned in the thymus, so in the thymus they're really being educated. So as long as they have learned in the thymus that when they see our own tissue, leave it alone. Do not respond to self-antigens. Only respond to foreign antigens. If they can do that, they're allowed to leave the thymus. So interestingly, out of the 100% of precursor T cells, immature T cells that go to the thymus, only about 5% leave. The rest are killed. And that is what tolerance is all about. They must learn to leave well alone self-antigens. So once they leave the thymus, they can reside in any lymphatic tissue, they can patrol around in lymph, they can travel in blood. B cells, they mature in the bone marrow, and then they leave and they go to these, the spleen, the lymph node, etc. Same. So that's the learning check, which I will let you do, and I hope this was very helpful to you. The third line of defense is very fascinating to study, so enjoy.